Hi there and welcome to this GCSE Biology Unit 5 virtual revision guide, and today we're going to be looking at communicable diseases, and this is quite a large module and some of the stuff at the end is only for separate science as it points out, so please be aware of that if you do trilogy. So we need to understand then what causes a disease. So communicable disease is a disease that can be passed from one person to the other. It can be transmitted. And transmission is caused by pathogens. And pathogens are microorganisms or microbes which cause diseases, so such as bacteria and viruses. The two main ones we need to know about are bacteria, which reproduce rapidly, as you know, so they split into two very quickly, and they make us ill by producing toxins, and they make us feel very poorly. They actually live on the outside of our cells, not on the inside, but they release these poisons, these toxins that make us very ill, and that damages our cells. An example of a bacterial infection, there are two main examples that we need to know about that can affect humans. The first type is Salmonella. Now, Salmonella is food poisoning effectively, so you need to know that Salmonella is caused by bacteria. You need to know it is caused by contaminated meat generally, and that is basically food that often hasn't been cooked, and it can also be spread through poor hygiene. And you can see the symptoms there. Just think of sickness and food poisoning if you ever ask about Salmonella. So fever, abdominal cramps, diarrhea, vomiting, sickness, those sort of things that you would expect. And the other bacterial infection you need to know about is gonorrhea. Now, gonorrhea is a sexually transmitted disease that is spread through unprotected sex. And the method of avoidance is one that you're expected to know for GCSE, that is by using condoms and protection or reducing the number of people you sleep with. And that is the main method. The treatment for both of these would be antibiotics. In theory, Salmonella obviously isn't going to last that long because food poisoning generally passes in a few days. But gonorrhea is a disease that if you don't get treated can get more serious. The symptoms that you need to know about are that you would get thick and yellow or thick and green urine or discharge, and that then causes pain when urinating in both males and females, and it can also cause inflammation, and it can also lead to more complications later on. Now, viruses are different, as we're obviously much more familiar with viruses these days, but a pathogen obviously is a microbial disease. A virus is actually small enough to go into your cells, and it goes into the cells and needs to go into the cells to reproduce inside them. Now, whereas bacteria we can kill with antibiotics because they're on the outside of our cells, viruses aren't so easy to kill because they go onto the inside of our cells, and as they reproduce, they'll then damage and destroy the cells, often bursting out when the numbers of viruses get too high. And there are a couple of examples of viruses that you need to be aware of. Measles, okay, and the symptoms of measles can be anything from just a runny nose to a cough to loss of appetite to fever, spots on the skin, things like that. And as you get older, measles can have some serious complications as well. It is spread by it being a droplet infection, so coughing and sneezing. It can be fatal, and you will have most likely been vaccinated as a youngster in the MMR vaccine for this, the measles, mumps and rubella vaccine. HIV and AIDS is much more well-known illness, I suppose, and it's a sexually transmitted disease, again, spread through unprotected sex, but it can also be spread via use of needles for drug takers, or through mother to child, if the mother has HIV, because it's spread through body fluids. Effectively, we can avoid it by not reusing needles, by using condoms during sex, and things like that. Now, HIV is a virus that attacks your immune system, and eventually your immune system gets so weak that that your immune system can't cope, and that's when it becomes AIDS. So they're the same disease, and HIV stands for Human Immunodeficiency Virus. So it is a virus, and we cannot kill or cure the HIV virus at the moment. There's a third group that you need to be aware of pathogens called protists. And protists, the only example that you need to know about is malaria. Now, malaria is passed through what we call a vector, and the vector that spreads malaria is the insect, a mosquito, but it's actually only the female mosquito of a particular species, but you just need to know it's the female mosquito at GCSE. Now, what happens is then the protist enters the blood when you're bitten by the mosquito, and it moves to your liver, and there it basically it reproduces, and that can cause you all sorts of problems. So it can be anything from headaches to fatigue to vomiting to sickness to chills to fever, and obviously a lot of people die annually from malaria. Now, prevention of malaria is easier than a cure. So the best way to prevent malaria is with taking drugs if you go on holiday, and you have to take those before you go on holiday to make sure that the mosquito, the malaria can't get into your bloodstream. You need to be able to, you could potentially remove all the mosquitoes for trying to kill them off in certain areas, drying out swamps where they reproduce, for example, or you could use nets which you put over the beds at night time, because that's the main time that they are active. We also need to be aware of plant pathogens, and please don't overlook this because some people often think, well, it's a bit boring, I'm not going to look at this. There are two main plant pathogens you need to really know about. The first one is a fungal disease. So funguses in humans might be athlete's foot or ringworm or something like that, and they're generally skin irritations that you can use fungicide to treat. In plants, fungus example is rose black spot. And as you would guess, it leaves black spots on the leaves, as you can see there. And basically all plant diseases have the same symptoms that you could really deal with learning and linking together. So basically, because the leaves are less healthy, and in this case there's black spots and they might go yellow, as you can see in the picture, the plant can do less photosynthesis. If the plant does less photosynthesis, it's going to produce less glucose, and therefore there's going to be less growth, and flower growth will be poor as well, and therefore the plant won't survive as well. It is spread, fungus diseases are, through wind and rain, and we can treat it by either burning the plants that have been affected to try and stop it spreading, or using fungicide. An example of a virus that you need to know is tobacco mosaic virus. And as I said a moment ago, the effects really are the same. It doesn't matter what the disease is. As you can see there, the leaves are affected, so you've got yellow leaves, so again, less photosynthesis occurs. If less photosynthesis occurs, less glucose is produced, less starch is produced, and therefore there'll be less growth, and the flower health of the plant will be lower as well. Now, tobacco mosaic virus is spread by direct contact, so it could be that two plants growing next to each other might spread the disease one to the next one, or through animal vectors, so stuff like insects travelling from plant to plant might carry the disease unknowingly on their wings, let's say, and pass the plant on. The treatment is to try and breed resistant crops, and also generally just trying to use better crop hygiene, which means that you try and avoid spreading it as the farmer or gardener. So then, how do diseases spread? Well, communicable diseases are contagious and are transmitted between people. So we're mainly focusing on people here, and there are lots of different ways diseases can spread. And you always need to try and link this to the disease. So don't automatically say coughing and sneezing. If it's a disease like sickness, that'll be through contact or through not washing your hands and things like that. So basically, diseases can be spread from airborne transmissions or droplet transmissions, so coughing. It can be spread by contact. So if you've got the flu and you shake someone else's hand, that could spread it. It could be spread through feces, through not washing your hands properly, to go into the toilet. It could be spread through contaminated water. And an example of that is cholera, which used to be a problem in the UK and it isn't anymore since we've developed good water cleaning systems. But it's still a problem. You sometimes see it on the charity adverts where they talk about buying filters to stop cholera passing in the water. Infection caused through by pathogens, obviously, from one to another. Then you could also have food, so it could be animal vectors, like in the picture of this person eating a burger that's got a fly on it, a fly's landed on it, and it's passed on that virus. Other animals, like mosquitoes and dogs and things like this, may bite, and that may pass on an infection. And it might be simply the food isn't cooked as well. So lots of different ways that diseases can be transmitted. So the primary defence mechanism is the natural ways that our body stops pathogens entering. So for example, here we've got some examples that you need to know. The skin itself is a natural barrier it's a water-resistant barrier, waterproof barrier. It contains antimicrobial secretions, and that stops bacteria being able to get in. If you obviously cut yourself, your skin or your blood will very quickly clot, and that means that it will stop an entry for the bacteria to get in. In our throat, we have cells called cilia or ciliated epithelial cells, but they have cilia on them, and we have mucus-producing goblet cells. So what these do is they produce mucus is naturally made to line up in your throat. Okay, the mucus then captures bacteria or viruses as you breathe in, and then the cilia then waft the mucus up your throat to the back of your throat. So it comes from into your airways, up your throat to the back of your throat. And then that allows you then to swallow it, and it takes the bacteria down to your stomach, where the stomach acid kills the bacteria. So you have cilia, which are little hairs, mucus in your throat, which captures the bacteria. The cilia then waft the bacteria back up to the top of your throat, where you swallow it. That takes it down to the stomach to be killed by the hydrochloric acid, which is the stomach acid, and you must say it kills the bacteria. Other things that we have are not necessarily you want to call them enzymes, but we have antimicrobial secretions in the eyes and in wax in the ears, which prevents us getting infection. We've got effectively other things around our body which help us, for example, our stomach acid, and that will do really. Unit 5 secondary mechanisms are white blood cells. So we have white blood cells in our which make up our immune system, and they can kill pathogens in 3 ways. The first way is something called phagocytosis, where you can see here the big white blood cell called a phagocyte. It effectively is engulfing and then digesting the pathogen to destroy them. So you don't say eat, because it's not eating it for nutrition. It engulfs it or surrounds it, and then it uses enzymes in the cell to digest it and destroy the pathogen. Second thing that the white blood cells do is something called a lymphocyte. That word is there for you if you want it. The lymphocyte produces antibodies that are specific to a particular pathogen. So for example, if you caught measles, let's say, your body hopefully would eventually produce measles antibodies to destroy the measles virus. So what the antibodies do is they kill the virus or the pathogen by either blocking their antigens, which are the little receptors on the surface of the cell or virus, and they can clump the pathogen together to stop them being able to work. And the third way which we use our white blood cells to destroy bacteria or viruses is we produce something called antitoxins. So earlier on we said bacteria release toxins, which are poisons that can damage our cells. So we produce antitoxins which counteract the toxin. So what if our bodies aren't doing everything we need them to do? Well, one method is to try and take drugs, and the type of drug that has been massively used in the last 50 years is antibiotics. Now, antibiotics kill bacteria, they cannot kill viruses, and this is for 2 reasons. One, viruses hide inside our cells, so we have to get into our cells to damage them, but also they're made of different things, and antibiotics damage the bacterial cell wall. They damage something called peptidoglycan in the bacterial cell wall, so they are very specific to damaging bacteria. They were discovered in the 1920s by Alexander Fleming and were later developed by the Pfizer organisation and obviously have had a massive impact. And what Fleming discovered was that if you have some antibiotic, and the one he discovered was penicillin, and the penicillin there is the white bit, you will get around the penicillin a zone of inhibition. So there'll be an area where bacteria cannot grow, and he realised that that must mean that this chemical, this penicillin, killed the bacteria. And then we started producing these in the Second World War, and then it's been used a huge amount in the last 50 years. Now that has led to a problem, and this is something you'll look at in more detail later on in the course, but it has led to antibiotic resistance. So antibiotic resistance is when some of the bacteria have become resistant to the drugs, so they're no longer killed by our antibiotics. And this happens because what might happen is if you have, look at picture number 1 there, lots of bacteria, you give them the antibiotics kill off most of the bacteria in the body, but then there might be 1 or 2 that are resistant, and because they reproduce asexually, then they will reproduce really quickly and they can spread, and bacteria can even swap genes between them as well to make it spread quicker. Now, why has this happened? Well, it's happened due to people not finishing prescriptions. So you might go to the point where you would have actually killed off the resistant bacteria, but you finished the prescription because you felt okay, and it gave those bacteria a chance to then reproduce and spread. We've overused antibiotics, so people used to get antibiotics for all sorts of different diseases, including viral diseases, which wouldn't have actually helped. We use them in our food industry with growing cattle to try and reduce their illnesses to make them grow quicker. We use them a lot of different places, and that has caused a problem. And now, to reduce resistance, doctors are using prescriptions with multiple antibiotics types. So if one antibiotic doesn't kill the bacteria, hopefully the other type will. You are also being told and instructed to make sure you complete the prescription, and also we're trying to reduce the usage. So antibiotics are used for bacterial diseases, but often with viral diseases we try and prevent getting them, because viral diseases are more difficult to kill, because antiviral drugs which are available often give nasty side effects because they actually damage our cells to get to the virus. So one way we can treat or try and prevent viral diseases is vaccinations. And vaccines work by injecting a small amount of the pathogen, and it's either a dead or a weakened form of the pathogen, into the body. And that's the key thing. That very small amount then makes your body and your white blood cells think that you've got that illness. So your body then creates antibodies to fight the disease, and if you ever actually get the disease in the future or get exposed to it, then that pathogen will be destroyed before you even know it. And this graph is a quite common graph. This sort of shows you what happens when you just get an illness, but it also could show you what happens when you get a vaccine. So if you get a vaccine, and if you know about the COVID vaccine recently, a lot of people got a bit poorly after having the COVID vaccine. And that is because what happens is it takes a few days for your body to produce the white blood cells and the antibodies needed to fight off that infection, and that therefore your body might feel quite poorly. As the antibody numbers go up, then you feel fine, and that's because your body's using a lot of energy to produce those antibodies. Then what will happen is those antibody numbers will drop off, but you'll have a few what we call memory cells. And then if you actually get exposed to the real virus or the real pathogen at some point, then you've already got the instructions to make those antibodies. So you can make them much more quickly. So if you look at that, it's much more rapid and a higher number of antibodies. So you need to know the secondary exposure, you produce a higher number much more quickly, and also it doesn't level off so quickly, it doesn't go down as quickly, it stays at a higher level afterwards, so you don't feel any illness. But we are always trying to find new drugs, and this is obviously very important. So to make effective drugs, what we need to do is look at what properties drugs need. So drugs obviously need to be effective. They need to work. They need to actually kill the pathogen and hide the symptoms if possible. They need to be safe and they need to be not toxic. We need to have as little side effects as possible because obviously they're to try and make you feel better, not worse. They need to be stable, which means they need to be able to be stored for a long time. And they need to be easy to take and also need to naturally leave the body. So after a while, when you don't need them, they're not still there adding up or causing problems. So effectively then, drugs are developed using a series of stages. And often initially we look at natural stages to find drugs, and often in plants. And two examples you need to know are that the willow tree was used as the initial product for aspirin. So that's where we developed aspirin. And plants that you might have in your garden, foxgloves, contain a chemical called digitalis, which helps heart disease. So you'd be expected to know what those two plants have produced for us. And here we have the stages of drug testing. So the first stages are what we call preclinical trials, which are the ones that don't involve humans. So once we've found our chemicals that we want to use, what scientists will do is create computer simulations. And what the computer simulations do is test different scenarios to see how well that chemical would stand up. We then may go on to actually cell testing before we go on to animal testing, where we'll look at cells and tissues. And that is really important because it's not considered as an ethical issue because you're just looking at a couple of cells, which obviously don't have efficient feelings. Okay, once you go past that, we go to animal testing, where we look at particularly, in some cases, do the drugs work, but we also look at the side effects on animals. And the small animals start off with, and then depending on the drug, it might get to larger animals. We then go to something called clinical trials, which is when we test on humans. And phase 1 of clinical trials would be to use healthy volunteers. And obviously all these phases, we start with low numbers to start off with, and then the numbers would increase. And healthy volunteers, we're not looking at does the drug work because these people are healthy. We're looking at does the drug have any nasty side effects? Is it safe to use? And often they'll use like students at university and pay them money to do that. Phase 2 then, we'll use a small number of people with the illness who are maybe otherwise relatively healthy, and we start testing for effectiveness and side effects. And then stage 3, they test larger numbers of people with the illness and try testing about how effective it is, what the prescription sites should be, and also are there any side effects. The drug will get licensed, and then again it has to be retested regularly. Now when we're doing these tests on humans, we use something called double-blind trials. And a double-blind trial is to see, basically we use something called a placebo test, where someone is given an empty drug or a pretend drug that doesn't contain any active ingredients. And when we use a double-blind trial, the doctor's giving out the drug doesn't know which ones are real and which ones aren't, and neither does the patient. And it's just to work out, does the drug show improvements due to their actual drug, or is it a psychological thing? It also avoids bias. Now, for separate students, you also need to know in separate biology about how we grow bacteria, to study bacteria in the lab to help us understand it, or maybe to diagnose diseases. So in order for bacteria to grow in the lab, they would need food. This is produced through agar gel, and it contains things like carbohydrates for energy, contain liquid water, it will contain nitrogen, which gives it protein, allows a bacteria to make protein. It contains oxygen, because bacteria are also grown generally better using aerobic respiration. It will contain moisture and water to keep the bacteria alive, and it will need to be warm. And the key temperature here is that 25 degrees if you're in a school is what we would use, because that avoids the growth of pathogens. Pathogens prefer, i.e. things that cause disease, are preferring to grow at 37 degrees C, the human body temperature. So in schools we keep it to 25 degrees C to avoid growing pathogens, to keep it safer for you. And what we use is something called an aseptic technique. And the key thing here is that this means that we try and keep everything clean to avoid you getting contaminated by bacteria, and also if we want to study a particular type of bacteria, to make sure no other bacteria get into our growth medium. So one thing we'll do is sterilise the Petri dish in the agar, and it's done by heat and pressure, and that will be done for you in class. You would then be expected to wash your hands because you will naturally have bacteria on your hands, and set up the equipment, and then probably wash your hands again. And then you will do something called inoculate, use the inoculating loop and sterilise it in a Bunsen burner. So you put it in the Bunsen burner, you might even put it in alcohol or ethanol, and then relight it again. So you know there's nothing growing on that Bunsen burner. You need to avoid contamination of the plate. So you see this person here doing a terrible version because they've just taken the lid off. That lid, as you hopefully should be aware, should always be at an angle. So you'd use one hand to do what that person's doing, and then one hand to keep the lid above it to try and reduce the amount of bacteria getting in from the air. We'd then obviously only grow the back of the plates at 25 degrees C to reduce the chances of human pathogens going. And once they've grown, or once we're finished, we'd keep them taped up. We wouldn't open them again until we wouldn't open them at all. So if you're asked to talk about a method for growing bacteria, the best way to sort of do it would say to do these things: sterilise the inoculating loop, and maybe add that into ethanol. Dip it into the bacteria you want to take a swab. Use a zigzag using the lid on as an angle, as I said before. So you must mention you'd keep the lid on at an angle to stop bacteria getting in from the air, and do zigzag plates across the surface of the agar. Replace the lid and quickly as possible, and add tape to it to prevent any extra microbes getting in from the air. And the last area that you need to know about is something called monoclonal antibodies. Now, this video is very long so far, so I won't be able to do much on this, but just really quickly, monoclonal antibodies are produced from a single clone of cells. So where we make an antibody that is specific to one particular antigen, which means that we can produce large numbers of one monoclonal antibody to maybe produce and use it in disease fighting, we'll talk about in a minute. So they will be specific to one antigen, so they'll always attach to one antigen. And you can see here how we make them. What they will do is they will vaccinate a mouse normally against the disease we're trying to make the antibody for. So let's say we're trying to make the antibody for the measles. We'd inject the mouse with measles. The mouse that would survive, we'd then take its spleen cells, and we merge them spleen cells with a tumour cell to make something called a hybridoma cell, and you need to know that name. So we get the antibody instructions from the spleen cell of the mouse. We get the fact that tumour cells grow rapidly, so they would produce lots of copies of this cell that would then know how to make the antibody. And the hybridoma cell will be grown in labs, and it would produce the antibodies that we need. So obviously there's ethical issues here because the mouse wouldn't survive during this, and many mice have probably died in producing this. The uses of monoclonal antibodies could be pregnancy tests, measuring the levels of hormones in our blood, we could be looking at drugs tests, things like that, identifying specific molecules that we're researching and looking at diseases maybe, or treating cancer cells. And the problem we've got is that often monoclonal antibodies have actually created a lot of side effects. So at the minute they haven't been as successfully used as before as we'd hope. So the last thing is then, well, what can we use them for? So one idea is we could use the monoclonal antibodies to try and kill cancer cells. So it could be that you either attach a drug to a monoclonal antibody. So we make an antibody which has got the shape that is specific to an antigen only found in the mutated cancer cells. If that will then only bind to cancer cells, we could attach the drugs that are used in chemotherapy to those monoclonal antibodies. And because they only attach to the cancer cells, those drugs used in chemotherapy would therefore only damage the cancer cells and not the rest of the cells in your body, which current cancer treatments can do. Another way could be that you could attach a chemical to that drug, which might send a signal to the white blood cells to direct them to attack those cells, which would mean your white blood cells could effectively kill off the cancer cells. Second mechanism we can use it for is we use monoclonal antibodies already to test for test for the presence of diseases such as HIV. So the monoclonal antibodies are used in this case to attach to HIV, HIV antibodies, and then we can use them to see, is that person's blood test, does it have HIV? And pregnancy testing is also used. Antibodies are used and monoclonal antibodies are used. And basically what happens with pregnancy testing is the lady urinates onto the pregnancy test. If you are pregnant, you produce and release a hormone called human gonadotrophin hormone, or HCG, I think it is, in your urine, and that attaches to monoclonal antibodies. The monoclonal antibodies then move up the stick, and if you're pregnant, then that produces a line showing you that you are pregnant.